شاء الله ان شاء الله. يلا أنس وين صرنا؟ خلصنا الباوربوينت الثاني صح؟ طيب يعني هون. طيب تمام. نكمل موضوعنا فيما يتعلق بالهرمون ميتابوليزمز او الهرمونز ميتابوليزم ان جنرال. راح نتكلم اليوم عن two examples mainly اللي هو الكاتيكولامينز and thyroid hormone, how they are made and how they are going to be metabolized. As you notice, the precursor molecule for both is the tyrosine, but the difference is in the pathway of the metabolism or synthesis in general. So all synthesized from tyrosine again. كل المركبات راح نحكي عنهم اليوم هم synthesized من one molecule, but one amino acid in general, and they have different localization as a matter of tissue specific localization, like a dopamine synthesized in the brain, as you know, as central nervous system, norepinephrine synthesized in the brain, as well as the adrenal medulla, specifically in the chromaffin cells. الابنفرين methylated norepinephrine بمعنى آخر هو عملية methylation للنورابinephrine norepinephrine actually, and represents 80% of the catecholamines in the medulla of the adrenal gland. So this is the methylation process. We are converting the norepinephrine to epinephrine by addition of the methyl group through the synthetic pathway that you are going to learn today. These are the four major steps of the synthesis, starting with hydroxylation process, decarboxylation, side chain hydroxylation, as well as methylation. So this is how the norepinephrine and epinephrine molecules are going to be synthesized, starting from the precursor molecule, who are tyrosine, by having actually coenzyme known as tetrahydrobiopterin, one of the most important coenzymes in our metabolism of the nitrogen-containing compounds. And the action is conducted by the tyrosine hydroxylase enzyme. So once it's hydroxylase enzyme, it means that we are going to add hydroxyl group here on the ring itself of the tyrosine. So it will be converted to 3,4. معنى ذلك إن البوزيشنز للهيدروكسيل جروبس صاروا على هذول two carbons. دايهيدروكس أو دايهيدروكسي فينول أنين إحنا بنسميه دوبا مولكيول اللي متعودين عليه. هلا هذا الستيب بسموها rate limiting step of the catecholamine synthesis, or الكاتيكولامينز اللي هم كل مركبات نتكلم عن الدوبامين الدوبا as well as سواء كان norepinephrine ولا epinephrine. But that's why they are called catecholamine. So this is the rate limiting step, which means that if this step is going to be stopped, we will not have any other future molecules. ومن خصائص الريت limiting step in general, it's irreversible step, regulated step, and it's the only step that is going to produce these products. So it can be inhibited. We could have deficiency in these or in the tetrahydrobiopterin, or sometimes we could have deficiency in the recycling of the tetrahydrobiopterin. So we have different causes of the deficiency of the catecholamines. Good. مضطرين نضل هيك. طيب. Again, deficiency بالcatecholamine synthesis could be due to the mutation in the enzyme, basically. وهذا very easy to know. Sometimes it could be deficiency in the tetrahydrobiopterin itself, or deficiency in the recycling of the dihydrobiopterin. لأن عملية الهيدروكسيليشن بتحول للتترا إلى dihydrobiopterin molecule. Or it might be the enzyme that is involved in the recycling of the tetrahydrobiopterin. فالأمر مش متعلق فقط بالإنزيم المسؤول عن الريت limiting step. It's also related to the availability of the coenzyme in this example. So this is the first step again, and the second one is conversion of dopa molecule to dopamine by decarboxylation reaction. Since it's decarboxylation, so the enzyme is called dopa decarboxylase enzyme. And I think some of you for sure remembering the pyridoxal phosphate molecule as a coenzyme involved in every decarboxylation reaction. لما بنشوف decarboxylase enzymes, usually they rely on the action of the pyridoxal phosphate molecule. ليش هذا المولكيول مهم جدا as a coenzyme؟ لأنه المسؤول عن the transfer of carbon molecule outside of the molecule, or releasing of the carbon molecule as a carbon dioxide from the original molecule. So the dopamine next step إذا بنتبع الأربع خطوات اللي حكينا عنهم, it will be hydroxylation by hydroxylase enzyme. And again, here we need some coenzyme like copper as well as ascorbic acid. هذا بذكرنا بالcollagen metabolism. They have the same components when we talk about hydroxylation of the collagen prolines or lysines. Okay, it's similar to the same reaction. So eventually you will have norepinephrine, and by one more step you will have addition of the methyl group to the molecule. So it will be converted from norepinephrine to epinephrine by methylation reaction. Okay, and the coenzyme that is involving this step is actually the enzyme is called phenylethanol-amine methyl transferase enzyme. المثل هون مش مبين, لازم تكون موجودة. So I will show it in the next slide. So yeah, it's here. We have S-adenosylmethionine as a famous molecule that is involved as a coenzyme as well. So every single step in the catecholamine molecule synthesis, we need certain coenzymes because we are conducting transfer or hydroxylation process. Transfer معناها الكربون انت بتضيفها او بتشيلها. Hydroxylation process involves addition of hydroxyl groups to the eventual molecule. So collectively, the tyrosine will be converted to either epinephrine or norepinephrine by the steps of hydroxylation, decarboxylation, and again we have methylation, hydroxylation, methylation in the final step. So you have to memorize all pathway of the catecholamine molecules. هلا مين الكاتيكولامينز اللي احنا دائما بنعملهم measurement؟ dopamine, norepinephrine, and epinephrine. So you have to know the differences between these molecules. So basically, they are derived from single amino acid known as tyrosine. One of the things that you might know, اللي هو Parkinson disease, as a neurodegenerative disorder, is due to insufficient dopamine production in these individuals, and result in what's called idiopathic loss of dopamine producing cells in the brain. And administration of L-DOPA as a treatment, like a treatment of levodopa بسموه, is the most common treatment in order to compensate the loss of the dopamine molecule in these individuals. The other two hormones that can be derived from tyrosine, but in different tissues, because we have different enzymes and different localization as well, لهم T3, T4 hormones, tyrosine hormones. هلا حكينا عنهم سابقا briefly انه they are synthesized in the thyroid gland, and we have processes of iodination and organification process. So today we are going to rediscuss this issue starting from the beginning. So first of all, we need to what's called iodination of the thyroglobulin protein, which is synthesized by thyroid cells. And this thyroglobulin protein is going to be iodinated on the tyrosine residues. And here we have a certain kind of disease related to the Goiter disease, related to the iodine deficiency, and then thyroxine deficiency. And the thyroglobulin protein is going to be entered to the cell actually by the pinocytosis process rather than phagocytosis. So it depends. بس بالنهاية هي عبارة عن endocytosis called pinocytosis, since you are taking small solutes or small molecules. Lysosomal digestion will break down the thyroglobulin and release the thyroxine hormones in the active form. So 30% of the hormones are going to be released to the extracellular site, to the blood circulation, I mean. And we have like the rest is going to be deiodinated and recycled again and again as a protein of thyroglobulin. إذا بترجع للسلايد اللي قبليه, this is the continuous process of the releasing of the iron and resynthesis of the thyroglobulin again in these cells. هلا الانتي transportation of the thyroid, sorry, iodine in the cell is ATP dependent process. Indirectly, I mean, and it needs transportation of the sodium outside of the cell as a antiport mechanism of transportation. هلا بتلاحظ الجويتر disease is associated with many deficiencies like deiodination, for example, transportation of the iodine, or even the excretion of the iodine or the action of the peroxidase enzyme. So we have different conditions that might be associated with the goiter disease that is related to the iodine availability. هلا الثيروغلوبولين basically is large pro-hormone. هلا pro-hormone في عليه conservation يعني بس because it will be cleaved to produce the hormone, that's why it's called a pro-hormone. Pro-hormone المعنى الحقيقي لها you are cleaving the molecule and you produce hormones from this molecule rather than modification of certain amino acids and cut of these amino acids. So it can be called pro-hormone molecule. We call it scaffold protein actually. It's large iodinated one and it's glycosylated protein or glycoprotein basically because the percentage of the carbohydrates that's found in this molecule is like from 8 to 10, the iodination percent actually. And the iodination or the iodine molecule in the thyroglobulin represents 1% up to 1% of the molecule, composed of 2 large subunits and contains like 115 residues of tyrosine that are potential for the iodination in order to release the thyroxine hormones, and mainly we have percentage of 7 to 1 according to the reference that we have. طيب هلا كل هدول بيتعلقوا بالايودين or iodide metabolism. So the thyroid is able to concentrate the iodine against strong electrochemical gradient due to the active transportation that we saw before. So there is a potential or a capability to store the iodine in the thyroid glands because it's dependent on the ATPase pumping system, and the ratio of the اللي هو T to S ratio in the human اللي هو thyroid to stimulation factor بسموها أو ratio in a human is on normal iodine diet is about 25 to 1 stimulated by thyroid stimulation hormone as an active form of the thyroxine hormone. كل الاشياء هاي اعتقد تعرفوها بالفسيولوجي يعني فيش داعي ارجع اعيدها لان اصلا الوقت مش مساعد. هذا الموضوع ناقشناه سابقا اللي هو cleavage of large hormones for example or pre-pro hormones in order to produce the active site like insulin, thyroid hormone actually or parathyroid hormone is also released as a large peptide that's going to be cleaved at different sites. So just to know that we have this example as a pro-hormone. So that's why we call it pro-hormone, because we cleave the polypeptide in order to have the active site of that or the active molecule of the hormone itself. So this is not a human one, it's bovine pre-parathyroid hormone as an example. مش بالضرورة الحفظ, just to know that we have a cleavage sites in order to activate the hormone. ال PTH synthesis مش راح أخش في التفاصيل تبعته لأنه just to know it's released from the parathyroid hormones. We have these بسموهم cleavage enzymes اللي هو cathepsins like cathepsins B and D. They are involved in the parathyroid hormone synthesis and they are responsible for the cleavage of these sites. Angiotensins, as you might know, like angiotensin 2 is also synthesized from large precursor molecule as another example by cleavage process. Just to know that this kind of hormone is also pre-pro hormone molecule that is going to be generated from the angiotensin gene, and you are familiar with this pathway or this axis actually اللي هو ACE axis. Right. So we have this common enzyme اللي هو is enzyme that is responsible for the activation or synthesis of the angiotensins. We have also what's called opiomelanocortin hormones or peptides that are going to be generated by the cleavage of the pre-pro hormone molecule and having the other active forms. المطلوب منكم بهذا الجزء فقط معرفة انه type of metabolic pathway that are going to form these peptides or these hormones. They are peptide hormones actually. Okay, so يعني سريعا المطلوب نركز عليه بهذول السلايدز الميتابوليزم of catecholamines as well as الثيروغلوبولينز and thyroid hormones. هلا ما تبقى عبارة عن just cleavage processes to generate the active peptides that you already familiar with. طيب هنتكلم عن هذا الجزء أخلصه اليوم لأنه بدي أريحكم أنا من ما بعرف إذا رح ناخذ آخر محاضرة نلحق ناخذها ولا لا. أشياء تكلمنا عنها سابقا فقط للإعادة اللي هو signal transduction story. هلا السؤال ليش بنعيدها؟ لأنه يبدو في خلل بتوزيع المحاضرات فعمالهم بيكرروا نفس التوبيك في أكثر من مكان وما صح لنا وقت نشيل عدد من المحاضرات لسبب إنه راح يصير في خلل في التوزيع فقط. فإحنا مضطرين نحكي عنهم بإعادة بسيطة. إنه السيجنال ترانسدكشن اللي تكلمنا عنها مسؤولة عن transducing the signals from outside to the inside of the cell in order to face certain challenges that the organism actually could face or the organ could face itself. معنى آخر في بعض الorganisms like the microorganisms they are responding to